Polymerase chain reaction is based on the mechanism of DNA in vivo replication.
PCR takes place through cycles, each cycle consisting in 3 steps. 1. DNA denaturalization to single-stranded DNA. 2. Alignment of the pair of primers. 3. DNA synthesis through the DNTPs addition by the polymerase. There are different PCR variants, for example, nested PCR, which is used 2 pairs of primers, the secondary pair hybridizes to the primary PCR product.
In hot start PCR, the DNA polymerase is contained in an antibody, which will be inactivated until a certain temperature is reached, thus starting the amplification. Reverse transcriptase PCR is based on a mechanism used by retrovirus, producing DNA from RNA. This PCR needs one previous step using reverse transcriptase to synthesize complementary DNA that could be used as a template in a normal PCR reaction.
RT-PCR consists on a constant fluorescent product collection of a number of cycles and conversion of the fluorescent signals into a number which corresponds to the number of products obtained from the number of cycles.
Multiplex PCR consists on the detection of different microorganisms by using different sets of primers in the same reaction. Each primer is pathogen specific. And colony PCR uses individual cells that can be either lysed in water or added directly to the PCR reaction.
PCA technique is useful to synthesize large DNA oligonucleotides using a pool of oligonucleotides with short overlapping segments which determine the order of the PCR fragments. Droplet digital PCR consists of fractionating the sample in millions of drops which will undergo individual reactions to quantify precisely the nucleic acids for a specific sequence.
The following primers were designed for the amplification of EcoRIR gene and EcoRI methylase, which reside within plasmid RI13 in Escherichia coli strain RY13. Primers are recommended to be designed with more than 18 base pairs. The regions near the interest genes were rich in thymine and adenine, thus affecting the recommended percentage of cytosine and guanine. The team preferred to design short primers with an adequate GC percentage. The melting temperatures are also optimal since the difference between them is less than 1.02 Celsius degrees. In order to clone in an expression vector, it is also necessary to add the restriction enzymes to the primers plus aleatory sequences that will protect them from degradation. These added sequences will be digested by the chosen restriction enzymes when they are added to the plasmid.
The following primers were designed to hybridize the ZFX and ZFG genes, which are homologous genes on the non-pseudoautosomal parts of the sexual chromosome. The amplification of these genes and their further visualization in an agarose gel is one of the methods for sex determination. An alu element is inserted into the last intron of the FZX, so when PCR is performed, and node-generated primers are used, which amplify this intron from both genes, the resulting size difference is readily resolved by agarose gel electrophoresis. The presence of an X-derivative fragment serves as a positive control for the amplification and indicates a female origin, while the presence of both this and the shorter Y-derived fragment is a diagnosis for male. Sex determination with blood samples can be applied to areas such as forensics research in the gender determination of a suspect or a victim, and for further DNA analysis to determine identity. Results: electrophoresis gel with several runs of PCR products from human DNA, blank samples, and a positive control of previous PCR product in the wells 13 to 16 performed by our team. There is no clear evidence of the PCR segment amplification. The lack of amplification in the positive control well indicates several problems in the procedure followed. The blank indicates that there is no cross-contamination, indicating that the pipetting technique followed was the correct one. The thicker line at the bottom is interpreted as possible non-specific products.
When performing a PCR, it is important to consider the following.
Reagents, purity, and avoiding contamination. It is good practice to aliquot all reagents used for PCR into multiple tubes so that a fresh set of reagents may be used for each experiment. Also, Good lab technique and wearing gloves is crucial for the success of PCR. Choice of template. The template of DNA can be either chromosomal or plasmid. Primer design, complementary and specificity. Primers must be necessarily complementary to the region of the template flanking the target amplified. Length. Most primers are between 20 and 30 base pairs. It is important to consider the melting temperatures TMs to achieve a good PCR. Self-complementary and secondary structures are undesired structures in primers. Magnesium concentration usually is between 0.5 and 5 millimolar for the template set. Cycling temperatures are important for the denaturalization and amplification to take place during the PCR. The results of electrophoresis PCR products reflect an error in the procedure. The wells do not present contamination, which indicates a good control in the reactant distribution. The lack of results in both DNA well and the positive control indicate a problem while performing the PCR. The rest of the samples were processed in the same thermocyclator, which indicates the machine worked correctly. The error can be added to the PCR components such as the DNA polymerase or the primers when they were added to the master mix. If the master mix was not prepared correctly, the PCR will not take place.
Through the experimentation of this practice, the team was able to analyze both the ZFY and ZFX genes. The PCR results showed that the procedure was not followed correctly due to the several mistakes found, and in order to determine the sex of the person sample, these mistakes should be identified and corrected.